The initiation of replication requires a particular DNA sequence called the origin. This sequence is called ORIC in the E. coli chromosome. A protein called the initiator protein binds to ORIC and causes the DNA to unwind. Unwinding requires several proteins. The most crucial are DNA helicase, which breaks the hydrogen bonds that hold the two DNA strands together. Single-strand binding proteins, which help maintain the DNA in the single-stranded state. And DNA gyrase, which reduces torsional strain. An enzyme called primase synthesizes short RNA primers to allow DNA synthesis to begin. DNA polymerase III synthesizes DNA, beginning with the free 3' hydroxyl group on the last RNA nucleotide of the primer. Because the two nucleotide strands of DNA are antiparallel, replication takes place continuously on one strand, the leading strand, and discontinuously on the other, the lagging strand. The short lengths of DNA produced by discontinuous replication of the lagging strand are called Okazaki fragments. DNA polymerase I cuts out the RNA primer and uses 5' to 3' polymerase activity to replace the RNA nucleotides with DNA nucleotides. DNA ligase seals the nick between the DNA added by DNA polymerase III and that added by DNA polymerase I without adding another nucleotide to the strand. Through these steps, the replication machinery of the cell uses the parental DNA as a template to synthesize the leading and lagging strands.